first understanding, we will look at the second line of defense against infectious diseases. Another name for it is called the innate immune system, and the system has two very important characteristics, it being non-specific and non-adaptive. When we talk about the innate immune system being non-specific, it means that the system does not differentiate between different types of pathogens. When we talk about the innate immune system being non-adaptive, it means that it responds to an infection in the same way. And some of the components include having phagocytic white blood cells that will aid in the digestion and engulfing of foreign substances. Another component includes inflammation and high fever as part of the inflammatory response. So phagocytosis is a process where solid materials such as pathogens are ingested through endocytosis of a cell. And as a little reminder of what we learned in topic 1, endocytosis is an active transport that moves substances into a cell by enclosing them in a vesicle made out of plasma membrane. So in response to infections, phagocytotic leukocytes go through extravasation, which is a movement of cells out of the blood vessels and into the blood tissues. So when these tissues are damaged, they release chemicals called histamine to attract white blood cells to come into the site of infection through chemotaxis. The phagocytic leukocyte reads and recognizes a pathogen. Its job is to engulf and destroy the pathogen via phagocytosis. First, cellular extensions called pseudopodia surround the pathogen and fuse together to form an internal vesicle. Next, the vesicle is joined to a lysosome to form phagolysosome, to which afterward the pathogen is digested. The adaptive immune system is the third line of defense against infectious diseases, and unlike the second line of defense, this third line is more specific in its response to targeting specific and different pathogens. And to do so, the system has something called the immunological memory, where it is able to recognize and respond to specific pathogens upon re-exposure to prevent the development of symptoms. And it is because of this memory that the system is able to create a specific response to target the pathogen. So in order to understand this new line of defense, we need to understand something called lymphocytes. And lymphocytes help coordinate the adaptive immune system to produce antibodies. There are two main types of lymphocytes, which are B lymphocytes, also known as B cells, and T lymphocytes, known as T cells. B lymphocytes are cells that produce antibodies that will recognize and target a specific antigen, and T cells aid in regulating the release of cytokines to activate B cells. Now we need to understand the difference between antigens and antibodies. So antigens are substances that the body recognizes as foreign and will elicit an immune response. And the structure of an antibody is made up of B cells that are specific to the given antigen and have a Y-shaped structure due to their 4 polypeptide chains joined by the disulfide bonds. On the other hand, antibodies are proteins that are produced by B lymphocytes and plasma cells and are specific to the given antigen. Antibodies have different variable regions at the end of the arms where specific antigens can bind to them. Much like the relationship of enzymes and substrates that we learned in the previous topics, only specific antigens are able to bind and interact with their respective antibodies. So when phagocytic leukocytes engulf a pathogen, to stimulate the third line of defense, some present antigens on their surfaces. These antigen-presenting cells are known as dendritic cells that help process antigen materials to present to the T cells. When a phagocyte presents an antigen from a pathogen it destroyed, it hooks up with a helper T cell and moves to the lymph node. If the T cell reads the antigen and sees that it is a foreign substance, it releases something called cytokines to activate B cells that are capable of producing antibodies to come and help fight against the antigen. So the activated B cell will divide and differentiate to form plasma cells that can produce high amount of specific antibodies. So these antibodies latch onto the pathogens and it gives a mark that enhances the immune system's ability to recognize and destroy that particular pathogen. In the end, a small portion of activated B cells and helper T cells will develop into memory cells so that they can remember these pathogens to provide immunity in the future.